Thank you very much. Firstly, I want to thanks to the moderator for the introduction. And on behalf of the EDP Yogyakarta team, I would like to thank also for the committee giving us to the opportunity to present the update of our project in Indonesia. So I think Peter already explained a lot of things about the background of this project, including Wolbachia, and then how is we work with Wolbachia inserting to the mosquito, and then also some trial as well in some area in Australia, in Vietnam, also in the. We start to things to do as well in Indonesia, but it's quite different approach. So in this moment, I will present about the EDP Jogja, but we'll be focusing on the entomological aspect. So what actually we have been doing with the entomology, and then what the result, and then what actually our plan for release Wolbachia to the wild habitat here around Yogyakarta. So I think it's quite in line with the global goals that our program in Yogyakarta here is aimed to develop a natural approach to control dengue using Wolbachia. That's Peter already mentioned that Wolbachia is a common bacteria, safe, can be found anywhere around us. And yeah, this is quite a long project. We can see here that the project is divided to 4 phases, and then we just almost finished with the phase 1, which is assess the feasibility of this project to be conducted in Yogyakarta in Indonesia, and then prepare what actually the infrastructure, ability, on everything that we have to prepare for long of this project. And then the second phase is next year we plan to release in the small area, and then evaluated the result, and then thinking to broaden it in the bigger area. And the last project in the last of the project phase is how actually make it this project and this program become a national program or something, because as Professor Pratman said that it's quite challenging, it's quite difficult to control the Aedes aegypti, especially here in Indonesia. So yes, we started in September 2011, and then we just finished the phase 1. And then next, probably we're thinking next year we will entering the phase 2, which is the main component is release the Wolbachia Yogyakarta mosquito Wolbachia to the small area. So in Yogyakarta we work in the 6 areas, 3 sites in the Sleman and 3 sites in Bantul. So the area selected based on some criteria. First is a demographical character, which consists of the 300 to 1200 houses, and then this area must be surrounded by unsuitable area for Aedes aegypti. And then we have to monitor as well the entomological aspect, how actually common Aedes aegypti in this area, because we don't want to release the Aedes aegypti with Wolbachia in empty area of because the concept is release in small, as what Peter mentioned, that we release in the small number, expected to be mating with a wild one, and then transferring Wolbachia to the offspring of the next generation. So the last one is the most important as well, that the program cannot be applied without any approval from the community. So we are working intensively with the community, informing our program, and then we are now working with to get approval from the community how can we release in that area. So in terms of the release strategy, we will release the Wolbachia in 4 sites, and then using 2 another sites. So this is the area, and the red one is the targeted area. We will release the Wolbachia, mosquito with Wolbachia, and the green one is the area that we will use as a control. So about the phase 1, in terms of the entomological study or entomological activities, we did with many, with at least 4 activities. First is preparing colony, transmitting or transferring Wolbachia to the local mosquito through the backcrossing, and then maintaining the colony to the many generations until ready to be released to the habitat, the wild habitat. The second one is this is about the common concern that is it safe? Means Wolbachia is something that yes, on paper mentioned that Wolbachia is a common, Wolbachia is a safe, Wolbachia is environmental. I mean, it exists for a long time ago, and know it's still easy to be found around us. So the third one is examining the as Peter said that the Wolbachia has a capacity, has the ability to reduce the replication of the dengue. So we want to test actually how is their potential to reduce, and how is the rate, how is their potential. So the fourth one is monitoring intensively about the population of the Aedes aegypti in the targeted area. So first is about preparing the colony, Wolbachia. Yes, true that Wolbachia is Wolbachia is how actually we transfer the Wolbachia from that we bring from Monas to the local one. We do it we do it a back crossing for many times. Just to the aim is that we want to transfer the Wolbachia to the IKC of Yogyakarta Aedes aegypti in the projection that after 5 back crossing we will get almost 100% Aedes aegypti characterized with the local, I mean the Yogyakarta Aedes aegypti consists with Wolbachia. So it's already done. It's already conducted for 6 back crossing, and then already also maintained for 9 generation in the laboratory. It's quite happy that we got 100% of our colony consists Wolbachia. This is a quite stable generation to generation. So it means that we are ready to be released to the targeted area. So about the safety, this is quite important. So the common question is that how common actually Wolbachia in our area, and then second was will we release the strain that identical here in Yogyakarta, and the third one is that how identical actually the mosquito that we will release to the habitat genetically similar with in the local one. So first is about how common actually Wolbachia around us. We did wild insect survey. We catch it, we trap it, a lot of the different species of the insects. This is most, all insects come from the site that we are targeted to be a release area. And then the insect also come from, this is quite easy to find every day because it come from agriculture area, come from housing, in-house, and then also the insect that usually and there is in the food storage or something. So we already screened it 42 species, and then up to 50% sample have a Wolbachia. So probably less than what Peter said in the video. It's about 70%, something like that. But if we make it more sample to be tested, I think we will get a higher percentage of the Wolbachia there. So, but in this research, it's told us that the Wolbachia is really common because also Wolbachia found in the honeybee and what you call it, the Aedes albopictus and also Drosophila melanogaster as well. So the second one is how identic actually strain that we will release with a wild one. We collected a lot of the Drosophila melanogaster because the strain that we will use in the release is come from the Drosophila melanogaster. So we collected hundreds and then tested and screened it with at least a four primer that the primer can characterize how actually close, how identical one strain to another strain. So this is the result, and we can see that this is for what you call it, for primer that we use, the IS, PNTR, and PNTR as well, and then the line is similar. It's indicated that the strain that we will use, and this is and this is a Drosophila melanogaster come from the area that we will do a release, and this is Drosophila in Aedes aegypti that we are now storing it, we are now maintaining in our colony. So we can see that the strain of the Wolbachia that we will use is identical with what strain that we will use to release. So the third one is about how identical actually the Aedes aegypti. This is the common concern is that do we will release the new Aedes aegypti, or this is actually already exists there. So we tested how identical actually Aedes aegypti that we maintained in our laboratory with the white one. So we use microsatellite analysis, which is commonly used for mapping, for genetic mapping, for linkage analysis, and a heritage pattern as well. And this is can you wait? Okay. And from the result, it indicates that our Yogyakarta Aedes aegypti with Wolbachia is genetically identical with the white one. So I think Peter already mentioned a lot about it, about how actually we work with the vector competence to test the capacity of the Wolbachia in the Yogyakarta Aedes aegypti colony in blocking replication of the dengue. I don't want to repeat it, but just the basic line is that yes, Wolbachia in the Yogyakarta Aedes aegypti have a good capacity to block the replication of the Aedes dengue. So this is quite intensive working in the monitoring of the population inside that we targeted to release. We did monitoring on the adult population, larvae population, and also eggs population as well. And the first is about adult population. We use a PG trap. You can see in our board, exhibition board here. This is we set up 10 to 13 PG trap per set, and weekly sample or weekly collection. We weekly collected the sample. It's already almost 2 years, and then we can see from the chart here that Aedes aegypti is commonly found in all area, but different spatially and also temporally. So the peak population also usually occurred at the early of rainy season, but it seems quite different this year if we compare to the last year. There is like a big increase, but no, this is early wet season is here, but the population is still quite flat. So this is a spatial distribution of the Aedes aegypti in each area. we use the geospatial analysis based on the positive or negative orbit trap that we set up in each area. This is a sample inside it. This is a Trihango village. We see that the hot area, the area with the red color, is the area that the population of the Aedes aegypti. Along the survey, we usually find Aedes aegypti there. Different with probably in some area that only 1 or 2 times or 3 times or something, but in the hot, it means that what Aedes aegypti distributed clustered in each area. This is the same pattern as well, but we can see that more hotspot in the different area. It's not in the middle, but also in the on the end of the area as well. So this is quite interesting. What Professor mentioned about containers and the bak mandi or bed container as the breeding site. So because our curiosity is that in the dry season especially, it's difficult to find the container with water, but we still have a high population of Aedes aegypti in there. So then we did a survey in the well that how actually well as the potential of breeding site, and we find interestingly we found that 50% of the well can be a breeding site. So from the page 1, we can have summary that from the pig crossing we already succeeded to transfer the Wolbachia to the local Aedes aegypti. About the safety, we can say that the Wolbachia is the common bacteria. So we already deal with them, and this is a safe as well. And then Wolbachia that in the backcrosset Aedes aegypti is genetically identical with the local Wolbachia in the Drosophila melanogaster. And then there is a genetic similarity between the Aedes aegypti that we will release with a local Aedes aegypti. And in the Wolbachia as a virus blocking agent, we can see from Peter's presentation as well that yes, Wolbachia have the potential as the blocking agent for the virus replication. For the population of the Aedes aegypti inside, we can see that Aedes aegypti is a common insect that found in all hosts, have a random distribution with some cluster in the different area, and then also found that the well as a potential breeding site, especially in the dry season. So we are ready with we are quite confident with what we already got for the page 1, and then we will entering the page 2, which is the main objective is to demonstrate the establishment of Wolbachia in the wild habitat, and then how actually capacity of the Wolbachia in the anti-dengue properties. And then we already have ethical approval. We already have research permission as well. And then we are working with individual informed consent. And then with the support from the community, we will do a stepwise release. Firstly, we will release together in all sites, in the 4 sites. But firstly we will release in the 2 sites, and then we will evaluate, we will monitor, and then based on the result of the first release, we will continue to the next release. So we will release into first is in 2 sites, and then second is 2 sites, and then as a control site is 2 sites. So it's quite intensive working in the community engagement, as I told before, that this release on this program, on this project should be get approval from the community. We usually, we inform our project to community through the group meeting, female and male, and also one-on-one or home visit meeting. Our staff come to the host, talk to the community, inform about our program, get a feedback from them as well. And then what you can also be involved in the community event, for example, kind of like your Women Days, and we bring our equipment, our mosquito coming there, and then kind of like a discussing session with the community, and then informing our program to them. And then also we distributed communication material, brochure, calendar, EDP news, and also we put poster in each area. This is the aim is that we are transparently informed about our project to the community. So this is the last slide for ProEB. So during before the release, during the release, and after the release, we will do intensively monitoring on the Wolbachia. It's not only inside where we release, but also on the outside, as mentioned, as Peter mentioned, that probably it will be spread to the audit area. So we will do intensively monitoring on it, and also monitoring of mosquito population, how impact on the release in the increase of the population, and we will conduct the dengue screening, active case finding, vector competence to look how actually the Wolbachia capacity in the blocking of dengue after release, or probably after existing in the site for quite long time. And also we will do a safety surveillance in the environment, looking to the horizontal transmission of Wolbachia from our mosquito of Aedes aegypti to other mosquito. And also we will test the blood, human blood, to check is there any Wolbachia there. And the last one is monitoring the community consent, because again that every release we need approval from the community. So thank you for the team from the EDP, also from you all on the audience. Thank you.